Hello, everyone, and welcome to today's panel discussion, Adapting to Succeed, Perspectives from Leaders in Industry and Academia Using Flow Cytometry for COVID-19 Research. I'm Susie Valdez of Labroots, and I'll be your moderator for today's event. Today's discussion is presented by Labroots and brought to you by Sony Biotechnology. To learn more about our sponsor, please visit their website at www.sonybiotechnology.com. I'd like to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the discussion. To do so, simply type them into the Ask a Question box located on the far left of your screen and press send. We'll answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing this presentation, simply click on the support tab found at the top right of your presentation window, or report your problem by clicking on the Ask a Question box. So let's get started. As you know, the COVID-19 pandemic has influenced us all in many ways. The scientific community has really shown resolve to continue their work in the reality of this pandemic. Researchers around the world have come together to design antiviral vaccines, develop diagnostic tests, and set up testing facilities in their labs. They have found creative ways of donating their time, expertise, and supplies to accelerate the research and aid preparedness during this time. Today, we will be discussing with leaders from the industry and academia how they have adapted to succeed and contribute to COVID-19 research. Joining us on our panel today are Dr. Frederick Laduke, who is the CEO and co-founder of Immune Biosolutions, a biotech company focusing on the discovery and engineering of next-generation biologics. Dr. Laduke holds a PhD from the University of Sherebrook and has a background in biochemistry, biotechnology, and ecotoxicology. Our second panelist is Patricia Rogers, who is an Associate Director of the Flow Cytometry Core Facility at Broad Institute of MIT and Harvard. Patricia founded and has been leading the Flow Core for the past 5 years, where they support about 300 users and over 20 research instruments, including flow cytometers, cell sorters, and molecular biology instrumentation. Patricia has a background in microbiology and cell biology and has been in the field of flow cytometry for the past 15 years. And welcome to our third panelist, who is Dr. Jeff Milish, who is an Associate Professor of Medicine at the University of California, San Francisco, and the Director of the UCSF Core Immunology Laboratory that provides support for sample processing and immunologic assays required in basic and translational research projects. Dr. Milush holds a PhD from the University of Texas Southwestern Medical Center and has a background in molecular microbiology. Fred, Patricia, and Jeff, welcome to this session. So let's begin. This first question is for all of the panelists. We'll start with you, Patricia. Tell us how you and your team are doing and what the current status of your research or biotech facilities being open.
Hi, Susie. Thanks for the question and also for Labroots of having me as a panelist today. To start, as Susie mentioned, I run a large shared instrumentation facility or core facility at the Broad Institute. We're a group of 7, so there's 2 flow techs, 2 staff scientists, 2 admin, and myself, and we have a plethora of instruments. And we're all doing well. We're managing everything the best we can right now. And what we primarily do is we mostly train researchers or offer consultations, but we also do offer some staff-run services. And you know, there really were 3 phases of us being opened over the past few months. And so when I think back to early March, really the first week of March, the Broad held a town hall to have a discussion around coronavirus. I remember one of our Core Institute members highlighted the need to be concerned, but not overly worried. A week later, all of our admin and data scientists were sent home to begin remote work. Our 2 admins in our group transitioned that week to supporting us remotely. At that time, the rest of us left in the lab wondered how safe was it to be working in a core facility. Core facilities are, by their nature, hubs of connectivity and activity, and we can have up to 30 people in the lab at one time working. So that was kind of a little worrisome to all of us. And then that second week of March, the 3 remaining staff and I started working in paired shifts in the lab. The routine transitioned to staff and I working alternating days. And then during those days, we would do a lot of cleaning. So cleaning high-touch spots in the lab with bleach or hydrogen peroxide wipes and stocking the lab with supplies because we weren't really sure, are we going to be able to come back the next day? You know, are we going to be all healthy? So we were really just trying to prepare the lab in case we had to shut down. And it really felt like this mad dash in the facility because researchers were trying to wind down experiments because we all knew it was coming. And we were trying to determine which instruments should be left on and which others should be put in storage. So the third week is when we really got the notice that, hey, we were winding down, everyone wrap things up, that isn't critical. And at that time, I felt it was really important for my staff to start staying home at this week. And so I continued to come into the lab that week, and I started putting the really large sorters that were staff run only into storage mode. And then I was trying to figure out which instruments should I leave on for the researchers to continue the critical research. Lucky for us, about 99% of our researchers are trained to run the instruments themselves, and most of the instrumentation in our facility is easy to use and quite robust. This was key for allowing research to continue without having to be physically on site in order to keep the density low at the Institute. I also made sure to leave detailed notes around the lab for where extra supplies might be or how best to take care of things to avoid problems. For the next few months, we transitioned to working at home as a team and supporting the researchers who were doing the same by hosting webinars or assisting with data analysis. I was mainly helping the on-site researchers by donating whatever we had in the facility that could be used to help with COVID research. Just little things like centrifuges or reagents. And actually, one of the impressive achievements of the Institute was to turn our CLIA-certified genomics lab into a COVID-19 testing facility for the state of Massachusetts. We are now able to run up to 35,000 tests a day for the state with a turnaround time of only a day. And then the last phase has really been to return to Broad. So on June 1st, we started our ramp up. Just 2 of us on my team returned that initial week to turn the instruments back on and to get the lab ready for the researchers who were returning on June 7th. And again, I've split my lab, my group up into 2 shifts. So we basically span the full day of 8 a.m. to 8 p.m., working into 2 groups, because I can only have about 6 people in my lab at a time. And this means all scheduling needs to be centralized to one person, and we have to limit how many instruments are used at one time. Every day before we enter the building, we have to answer a health questionnaire, and everyone is required to wear a face mask at all times. There's typically about 200 to 300 lab workers at the Institute at once, and most of us are working limited hour shifts. And this is really to, prevent, people from congregating in the Institute or having to eat lunch here. We're really only supposed to come on to get our work done and then to go back home. And we're still cleaning everything. So that's a big part of what we do every day is we come in and we clean everything before doing quality control on all the instruments. And we've also had to move some instruments out into new locations. But the main key is we're all working together to stay healthy and maintaining our social distance of 6 feet apart, and that's been working so far for us.
Thank you so much, Patricia. And how about you, Jeff? How is your team doing? What is the current status of your research and your facilities?
Yeah, thank you for having me on today, telling you a little bit about what we've been doing. So my team and I are doing well overall. It's really hard to believe that we're already in mid-July and that this all started back in about mid-March. But I have to really commend the team on the great job that they've been doing. It's not that often that we get to talk about our team and the work that they do and how they support all these various researchers. But they've really done a fantastic job of adapting to the ever-changing demands and schedules that were needed to accommodate the studies. We basically began pivoting in mid-March to doing COVID-related research, and we had to shut down about 20 or so, 25 active studies that were recruiting patients and sending us specimens, or that we were performing assays on, and we really had to shut all that down and transition over to the COVID research. And in addition to kind of changing the types of studies that we were working with and sort of the types of biohazards that we were dealing with, there was just this increased stress of not really knowing what was happening on a day-to-day basis at the beginning, and trying to adapt to the new biosafety conditions that we had to work in, figuring out how just to commute between your home and work safely. And all of this was kind of being done, as I said, in the process of ramping down the ongoing studies and then increasing workloads on the COVID side of things. And that, in order to really meet those demands of the COVID research, we had to shift our hours. And it was very impressive. The staff were all very willing to change from their normal sort of 9 to 5-ish job schedules to basically running 7 days a week at the beginning. And at times that meant coming in as early as 6:30 in the morning and staying open as late as 7 o'clock or so at night in order to accommodate all the specimens that were coming in from the hospital. But I really have to commend the team on their willingness to adapt to this and their ability to stay focused on these tasks and provide our highest quality service that our investigators have come to know us for. And that's not easy given all the changes that were going on. In particular, my lab manager, Norm, did a fantastic job. He had the responsibility of combing through all these studies that were coming in, making sure that the protocols were in a format that the lab could read through, digest down, and be able to perform the assays and the processing that the studies needed. We had to make sure that basic supplies we could get our hands on in order to process the sample safely, including PPE, which at the beginning was definitely difficult to get our hands on. And then my processing team lead, Christina, she really did a great job of holding down the fort. Her and the 4 people that work with her on specimen processing, they needed to change their shifts. They needed to start early. They needed to work weekends. And they did this incredibly well. They were always very dependable, and they just never missed a beat in cranking out high-quality samples, which was really nice to see. And our admin staff, they were kind of forced into city mandates of having people that were not lab oriented had to work off campus. And so Meredith and Val had to start working very early on from home in about mid-March. And this puts a strain on things. Our team is very close-knit. We work together well. We're used to interacting on a face-to-face basis and in order to get the work done. And so there was a learning curve, but they really did a great job of also staying engaged and helping the team remotely. And sometimes, the best examples of some of their help probably comes in the form of just providing snacks and organizing our what we called socially distanced lunches for the team to try to bring in some food, because it wasn't always that easy to go out and pick up your lunch at the restaurants around here. And so, it was kind of a little team pick-me-up, and everybody got a chance to, take a break and have a nice lunch. And couldn't necessarily do it all together, but it was nice to at least be able to give the team members a chance to enjoy something special while they're changing their lifestyle up in order to accommodate these studies. And then Val and Meredith also had the important responsibilities of keeping things running behind the scenes and keeping our databases going and continuing to run the reports and analyzing data as we were acquiring it for the investigators. But overall, I think we were fortunate. We never had to shut down the lab, and it was really a great opportunity for us to help with many different studies. We're involved in 9 or so studies at this point, and it's been fun and challenging to work with all the different investigators. Now that we're into July, most things have opened back up at some limited capacity here at UCSF. And so even though many of the cores did shut down to to various extents. Most of them have begun to reopen as of mid-May and are running at various levels of capacity right now. I think the main thing is we're still trying to maintain social distancing and things like that to keep employees safe and keep the operations going, but also to be able to support the work that needs to be done for not only the COVID side, but now trying to restart those studies that had to be put on pause. And that's really kind of our next big challenge now, is how we overlay the studies that were ongoing before the shelter-in-place took effect with the studies that are now coming in quite rapidly with many samples, unfortunately, due to the increasing number of infected and hospitalized people. But you know, that's sort of the next challenge. We'll see how that goes. But you know, working with the investigators now to try to basically get their studies back up and running, but at the same time make sure that we set realistic expectations with them in order to you know, provide everybody with top quality service, regardless of whether it's COVID or not. So.
Thank you, Jeff, for sharing that information. And Fred, how about you? How is your team doing? And tell us a little bit about the current status of your research and biotech facility.
Yeah, so just to give the audience a little bit of background, so our company is located in a small university town called Sherebrook in the province of Quebec in Canada. So we're about 4 hours drive away from Boston, up north, very close to the American border. We were actually hit hard by this pandemic pretty much the same time as New York was, mainly because we have the equivalent of a spring break in early March. So a lot of our population traveled to the U.S., to Europe, to the Caribbean, and brought back more than just souvenirs. So the province of Quebec was hit hard, but the government on provincial level, but also on federal state, decided to take immediate measures. So we've been able to sort of flatten the curve, and so our position was we were in a very strong crisis at the beginning, but we managed to sort of go through it. It's never perfect, as you all know, but definitely we were lucky in that sense. Very early on, we were fortunate that our government understood that life science companies should be kept open. So we were considered an essential services, mainly because we were driving new diagnostics, helping hospitals with diagnostic, try to either design new vaccines or new therapeutics. So definitely our sector has been very active, although it was a challenge to stay open. So and in a sense, if there's a sector that could stay open while there's that kind of pandemic, it's definitely the life science sector, where we understand the risk, we can implement procedures and so on, measures that would limit the infection. So very early on in March, we decided to put every scientist that we could working from home. And we were fortunate in terms of facilities. Our labs are basically a bunch of small labs, one next to the other. So basically, we limited the number to one scientist per lab. So we were able to sort of keep the work going, start our programs of discovery for COVID-19, and pretty much managed to stay at 100% capacity. I would actually say that we were more at 120 or 130% of our capacity. Definitely very motivated in finding new solutions, interacting with the ecosystem, with the community, from locally and internationally, to sort of bring what we could to help hospitals and to do collaboration that would have an impact as soon as possible. So really, I think it was a really bad situation, but at the same time, we saw the community just rise to the occasion. So definitely, it was a difficult situation, and I heard Patricia and Jeff tell a little bit how they did it. We did very similar things, but at the same time, we were part of this. We are still part of that race to find a therapeutic, something that would protect the people. So we're still very active in that space.
Patricia and Jeff, this next question is for you. Describe for the audience how your institution and your team had to pivot to respond to the emerging needs to support COVID-19 research, and what measures did your facility implement to minimize individual risk while handling COVID-19 infected specimens? And Patricia, we'll start with you.
So in late April, I started to get requests to sort samples from recovered patients of COVID-19, mainly whole blood from the Boston area. And while the risk is quite low for this type of sample, we still don't know the full story. So I reached out to my EHS director, and we had a discussion and decided that it was best to treat these samples with additional caution. These patients had recovered, they no longer had COVID, but we still weren't sure what to do. So it's not quite a BL3, but something like a BL2+. So we determined the best course of action was to limit the number of people who have exposure to these samples by isolating one cell sorter in its own room. And, my room is one giant room, so I had to figure out how do I get one of the cell sorters out while social distancing and move it to another room across the institute while social distancing. So it took a lot of teams, including space planning and facilities, EHS, and also Sony. We had coordinated to move a Sony MA900 plus its biosafety cabinet out of the main flow facility into a small room across the hall. So this room had some nice features. It had CARAD access, so I could limit who was in there, and also enough space so I could put some smaller equipment like centrifuges for spinning down plates, post sorting. And then, I already had it in our calendar system, but I took it away so people couldn't access it anymore, and only gave it access to people who needed to sort these types of samples. And also, I had to think about, in general, anyone now working with unfixed human samples, we needed to move their samples over to this room. And in mid-April, it was really just the samples coming from recovered COVID-19 patients, because all other science wasn't going on at that time. And because of, the nature of cell sorting, you really want to isolate these samples in its own room, because so if anything happens, you can contain it much easier, and you don't have as many people around. Like if I had this, these samples coming into the main room, I could have, 6 to other people in there that could be potentially exposed to them. But we're also handling COVID positive samples at the Broad, but in the BL3 facility, which is not really in my jurisdiction. So we had to come up with a plan, and we're working on it right now because there are going to be more requests to bring human samples back to the Institute from the hospitals nearby. And we have to decide when the samples come into the Institute, what the safety level of them are, and then what appropriate facilities should they go to for processing. So if it's infected samples or postmortem samples, they should go to BL3, and if they're recovered patient samples, they can come over to the BL2. And also if they're donor samples coming from the hospital and they've been screened, then they can come into the main facility under normal BL2 practices, but if they're coming from the hospitals and they're not screened, then they need to go into our BL2 plus room that has this single sorter in it. And some of the things that we've been asking researchers to do is either to have their samples, the people that they're getting the samples for, tested for COVID, or if that's not possible, then doing PCR tests on their samples to confirm that they're COVID negative. But all of this is, a synchronization between all of the groups and EHS and my group at Broad to make sure that, we're keeping track of all the samples that are coming in and that we're making sure that they're going to the appropriate areas in order to get processed.
Thank you so much, Patricia. And how about you, Jeff? What measures are your facilities doing to implement and minimize individualized risk, and how are you pivoting around the need for supporting COVID-19 research?
Yeah, this is a good question and something that we have to take very seriously in order to keep the lab running and our team safe. So when we went into the shelter in place on March 16th, this really changed our operations. One, it meant that we had to shut down all our non-essential work, which was basically anything that wasn't SARS-CoV-2 or COVID related. And second, it really fragmented our team. And leading up to this, we were aware that this shelter in place could be coming at any point. I think it got accelerated a little bit quicker than we had thought, but as soon as we sort of saw the writing on the wall, we began to prepare ourselves that some of the team would have to work off-site, and we started preparing them with the tools that they would need to be able to continue that work at home, which in most cases was making sure that that the team had access to computers and computer screens. And so we sort of took care of our people that we could have work remotely from home. Hopefully they would be able to stay at home and isolate there and be safe. And then at the same time, there were plans here on campus put in place to begin health screenings for all staff that were basically entering the campus. And we began to wear masks all day long while we were on campus. And so basically over time, there's been some changes here and there, but basically our administrative team still continues to work remotely, and anyone that is coming onto campus has to be go through a health screening. So it's a centralized screening, and then you're given a mask to wear if you don't already have one, and you have to keep that on while you're working at all times. Within the lab, we've taken additional precautions, and so part of what we want to do is maintain a low occupancy level on campus in general, and so we're only at about 25% of our working capacity on the campus. And so, just the number of people on this campus has been trimmed down in order to keep space between people. And then in the lab, we can accomplish this because we have multiple sort of satellite rooms, and so the team can split up and work in different biosafety cabinets without having to be right on top of one another. And this has really allowed us to keep almost a full team in the lab at all times in order to keep up with the work. But inside the lab, everybody is continuing to wear face masks. And then we also require everybody to wash their hands as soon as they walk into the lab, just cutting down on any potential fomite transmissions. And then we, on a daily basis, actually multiple times a day, high-touch areas in the lab are sprayed down with 70% ethanol and kind of kept clean so that any exposures can be minimized. And then, like I said, we set up these smaller rooms for handling SARS-CoV-2 infected specimens. So for us, anything that is suspected to be SARS-CoV-2 positive in terms of blood samples has to be worked with in a BSL-2 room. And in there, our lab staff have our normal liquid impermeable lab coats and safety glasses, but we also require them to wear double gloves. And within the room, anything that is, any of the equipment that touches these samples, we decontaminate to the best of our ability. So it's either spraying it down with 70% ethanol or 10% bleach. And then we also soak our centrifuge buckets and things like that in order to remove any potential contaminants that may have gotten on that. So there's definitely a lot more cleaning that's happening inside the rooms and inside the lab in general. And then for samples that are coming in that are from the trachea, so for those patients who are on ventilators, we worked with our building engineers and our environmental health and safety group to basically repurpose a small room that we had with 2 biosafety cabinets in it, so that room was under a negative pressure, and this is considered our BSL-2 star room. And so, like I said, in there we're allowed to handle tracheal aspirates, and not only is the room under negative pressure, but we have also have some increased PPE that we wear in there. So here we're wearing back closed gowns instead of the front closed gowns. We double glove and wear sleep protectors along with our normal eye protection. But probably the biggest thing is that we also are wearing N95 masks when we work in that room in order to cut down any potential aerosolization that may happen. And And again, everything is continued to be wiped down with ethanol and with 10% bleach solution, and soaked in 10% bleach if we can, for things like centrifuge buckets. So that's kind of what we're doing in the lab when we're handling specimens. We, for the most part right now, are not dealing with cell sorting of highly infectious samples. Most of our sorting has been being done by magnetic isolation, and so that is a lot safer for us right now. We can do that inside of a biosafety cabinet without much aerosolization concerns. And so it's allowed us to continue to work with samples, enrich subsets, and then do single-cell genomics assays with those samples. On our cytometers, though, right now we're requiring people to fix all of their specimens, whether or not they're COVID positive or not, in 1% paraformaldehyde, and in order to inactivate any of the pathogens. And then we have a BSL-2 cytometry room that has 2 instruments in it, and those instruments right now can only be booked one at a time to, again, cut down on the number of personnel in the room. And then we have a cell sorter that's in our BSL-2 star room in an adjacent building. And in there, since that's inside of a biosafety cabinet, folks, if they want to sort blood specimens from infected individuals, that would need to be done in the BSL-2 star with an N95 mask. That sorter is in its own wing of the BSL-2 star, and so it's physically distanced from other folks working in there, and it's also inside the hood to cut down on any potential contamination. For more infectious samples like lung or tracheal aspirates, those would require for us to be sorted in a BSL-3, and there we have a MA900 that's in a biosafety cabinet. And in order to do sorts like that, that would need to be coordinated with some other labs that work in that BSL-3, but in there, individuals would have to wear full PAPRs, and we probably would be running those sorts for the individuals and for those investigators rather than having them access that BSL-3, since there's additional training that would require. But full PAPRs are in there, so hopefully that, on top of being inside the biosafety cabinet, will cut down on any potential risks that are occurring. I think the big takeaway from this is that we are trying to keep our social distancing in the lab, wearing face masks all the time, and trying to clean surfaces to cut down on any potential transmission that may occur. So.
Thank you, Jeff. Now let's move to the discussion of research studies. Patricia, we'll start with you. What type of studies are being supported by your core labs, and how do the researchers use flow cytometry for COVID-19 research?
So there are many COVID-19 projects ongoing at Broad, and one way we are helping in the Flow Cytometry facility is by facilitating cell sorting of immune populations. So we enable researchers by training them on the Sony cell sorters so they can purify or enrich populations for mainly single-cell genomics work, because that's a lot of what we do at Broad is genomics. The most popular applications include bulk enrichment for 10x genomics library construction, or sorting single cells directly into micro well plates for library construction for applications like SmartSeq2 before going on to sequencing. There are other applications that we use flow cytometry for, not just sorting. We also do analysis. So we do a lot of CRISPR analysis, looking at fluorescent protein expression. And one example is a team at Broad have been working on an interesting area to explore how to do more COVID testing. And so the Zhang lab, for example, has led efforts to create a rapid COVID-19 test using CRISPR-based technologies, and it's called Sherlock. And that will help to create some more diagnostic tools that people will be able to use at home. So these are just a few applications of what we're doing in the flow cytometry and facility, and a lot of what we do is we just train people to do this themselves. And so we haven't been training people lately because we have to keep our social distancing. So we're trying to figure out that next step and how to do remote training for everyone. but everyone that's already been trained before pandemic has been in the lab and using the instruments for these types of applications.
Thank you, Patricia. And Jeff, how about you? Tell us a little bit about the studies being supported by your core lab.
Yeah, so our core lab is a little bit different. We're a full wet lab. So basically what I mean by that is that we are helping individuals everywhere from processing specimens, cryopreserving them. We run assays for the studies. And so we're seeing samples straight from the clinics that arrive within 10 minutes of being drawn from patients to those samples that are being shipped in from outside locations for processing and assay work. I would say our studies break down into kind of 4 major categories. 2 of those are on the therapeutic side, where we're supporting studies looking at repurposed drugs like remdesivir or canakinumab, or the other side of the therapeutics is looking at kind of cellular and immune modulatory drugs. So there was one group that we were working with that was doing mesenchymal stem cell infusions to see if those stem cells could help alleviate some of the ARDS symptoms in the severely ill patients. And then the canakinumab, convalescent serum that's being infused in some patients. We're collecting samples from them to look at what is happening with their viral loads and their disease progression. And then we have some basic science studies where we're just looking at host immune responses. And so there are studies with different cohorts where we are collecting blood specimens, urine, saliva, feces. There's many different tissues. I think everybody's trying to collect as many varied specimens as they possibly can. Some of those are being used right away by different investigators, and we need to get those shipped out to those labs. Others are just being banked for downstream applications and for studies that haven't necessarily come to light yet. But there's on our side, with those studies, we're doing everything from the specimen processing and shipping out to also helping run cytoph assays on these samples or running single-cell genomics studies. So these are cases where we're taking like tracheal aspirates and enriching for CD45 positive immune cells out of those tracheal aspirates, and then performing single-cell genomics assays on them to look at transcriptional profiles of the immune cells that are coming from these patients who are on ventilators. And then we also have some studies that are just doing antigen-specific T-cell responses and looking at the immunophenotype of the cells that are in these patients with different degrees of disease progression, and looking at their antigen-specific T-cell responses to the SARS-CoV-2 virus. I think, trying to understand the difference between the serology and the antigen-specific T-cell responses is going to be important. These systems work together closely, and understanding both arms of that adaptive immune response is going to be important. And then lastly, we have one study that's kind of more focused on viral shedding. It has immune components to it as well, but in that study, it's really focused on how long individuals shed infectious virus, and so we're collecting lots of different specimen types there, again, running some cytoph and doing some SARS-CoV-2 viral loads in the lab to understand how much virus is in the different locations within the body and for how long you can isolate infectious virus from those locations to sort of help with decisions of how long somebody would need to isolate themselves before they were not infectious to family members or the community. So it's a pretty broad array of studies that we're supporting, and those are just studies that my group is specifically supporting. There are certainly many others that are happening across the UCSF campus, and collaborations between both other academic institutions and industry partners as well, which I think is really important in driving both vaccines and potential new therapeutics for those folks who are infected or to prevent new infections.
Thank you, Jeff, for sharing. Now, Fred, this next question is for you. Tell us about the technology offered by your company, Immune Biosolutions, and how it works.
Yeah, so as I mentioned, our facilities, we do have a couple of interesting platforms, but I think we're quite well known for hacking the immune system of chickens. This is really an amazing species to work with. Definitely an interesting immune system, and actually we do know quite a lot about this immune system. Actually, B cells, the B stands for bursa, which is an organ only found in birds. So definitely there's a history of knowledge on this immune system. So what we do is we try to take advantage of this unique immune system that derives quite a lot of diversity in terms of antibodies very quickly. In the case of COVID-19, definitely we were looking at this. So we have several platforms that we're using to generate our antibody candidates, from target validation, antigen design, up to biomanufacturing. So different parts of these platforms were used to drive these programs. So we have a platform that is using bioinformatics in silico design to generate 3D peptides, so to give the immune system a synthetic peptide, but with a 3D shape. We haven't used that technology for COVID-19 because we wanted to go faster, and the crystals were not available early on. So we use a very broad spectrum approach with our antigens. So like I said, we use mainly the chicken to derive these libraries of antibodies. We have two platforms to select the best candidates. Both have advantages and disadvantages for sure. We have a phage display platform, also a direct B cell screening approach that will show in a few slides. And both are combined with next generation sequencing and AI algorithms to sort of find the best candidates very early on and filter the sequences of antibodies that might be problematic for different reasons, immunogenicity, for example, manufacturing, and so on. And after that, surprisingly, chicken antibodies are easy to humanize once you know how. They have a very conserved framework. So once you have the right amino acids that you don't want to change, we can derive very early on these humanized formats. So all this is combined under our roof to generate antibody candidates for most of the time for oncology and infectious disease. We have a couple of programs in oncology that are a few months away from IND, and we have a CDF program that is in vivo validation. So we had quite a lot of experience with different viruses, different difficult targets, and we definitely applied all this knowledge to derive very quickly programs against SARS-CoV-2. So I can tell you about two things about this program. So we wanted to act quickly to generate the best candidates to start with. So we use our chicken approach for sure. So we immunize the animals, collect the B cells, and use our B cell screening approach. We also recruited convalescent human patients and used about the same approach, but this time with human B cells. So this is a typical assay that we do on a platform. So these are very small wells, what I would call nano wells, about 2.6 nanoliters. So we can put one B cells in each of these 100,000s of nanowells. And in those wells, we typically use either a specificity assay or a functionality assay. In the case of the COVID-19, we didn't have a lot of time to generate and test these newly designed functional assays. So we use a specificity assay to start with. We had two types of beads, as you can see on the screen. One type was fluorescent. These were MTBs, that was our negative control. and we had these bigger beads that were not fluorescent that were conjugated with different proteins from the virus. So we could actually see after a couple of hours if our B cells were secreting these very specific antibodies. So within, so again, very typical example with this well. After 1 hour, you see a small cloud of red fluorescence, and actually that's our secondary antibody that is able to recognize the presence of an antibody. And as you can see on the lower panel, after 16 hours, these antibodies are binding to the non-fluorescent conjugated with the proteins of the virus. So very rapidly, within a few days, we could find these what we call pre-validated antibodies. They were sequenced, all of them, several thousands of these sequences of these antibodies were sequenced, analyzed by our AI algorithms, and now we are testing different antibodies with different assays in vitro, in vivo, and combinations as well, targeting different parts of the virus and the spike protein. So very, very interesting to see how we could use our platform to answer very quickly within a couple of weeks, instead of several years as we typically do in oncology. So definitely, it's giving us the opportunity to accelerate research and test our platform to its limit, and it was very successful. So we're very happy about our progress.
Thank you, Fred. And this next question is for all of the panelists. Based on your experience with this new health challenge we face, what are some of the key takeaways you can share with our audience? And Patricia, we'll start with you.
Sure. So one of the biggest takeaways for me as someone who runs a large shared core facility is rethinking the nature of how we share. I'm a one large room and I have a lot of instruments crammed into this space, so I can get as many people in here and as much stuff in here for everyone to use. That isn't working out so great now because we have to be 6 feet apart, and so I really have to limit what's getting used at the same time. And usually I tend to gravitate towards putting everything that's similar together in one area of the lab, and now I'm regretting this decision. And actually what I'm doing right now is working on restructuring my lab layout, so now I don't have all the same instruments clustered together. And it will be easier to have all the instruments used if I intersperse like low-used instruments with more popular instruments. And so that's my next task for the next few weeks, is to basically rearrange my entire lab, which is not an easy feat. And then also, being forced to work remotely has taught us how to stay connected. So even though we're apart, it seems like we're one team working together. And, in the past, like Jeff had mentioned earlier that he's a tight-knit group, we're also a very tight-knit group because it's only 7 of us. So we're kind of on top of each other all the time and just checking in constantly. And having to, switch gears a little bit and try to use new platforms more heavily like Slack and Google Hangouts has really been instrumental in keeping us all informed of what's going on, so that way we can help all the researchers in the lab. And so I think going forward, I'm going to investigate more, and also with my team, to see what are other platforms and what are other tools that we can use in order to stay connected. But yeah, those are a couple of my takeaways.
Thank you, Patricia. And Jeff, how about you? Could you share a little bit about your experience with this new health challenge we face?
I think for me, there's probably, there's multiple areas, but I think there's probably 3 key takeaways that I would want to stress right now. One of those areas is really that this response to the pandemic has been very positive, and it's really kind of reemphasized the power of collaborations in science, and both the collaborations within the academic institutes, but also between academic institutes and industry partners, as well as even seeing the collaborations between potential competitors in the industry groups. It's been really powerful to see this, and I really hope that going forward, that everybody sees the benefit of this, and we can really sustain this down the road to move science along quicker and bring new therapies and prevention strategies to the masses, basically. I think the other area that this has really made me recognize is the importance of your team and your family in dealing with these stressors and these situations like this. We really need to take care of each other. Everybody really needs to, help each other get through these challenging times, and everybody deals with these very differently. So it's important that we all check in with our team, and that's not just your team at work, but your family team as well, just to make sure everybody's doing well and make sure that we're all communicating and getting the work done in a safe manner, and just keeping people as happy as you can under the conditions that we're under. And then lastly, I think this is really similar to what Patricia said in that thinking about the layout of the lab, we were really fortunate in this situation that our current space, we were able to kind of move things around and reconfigure quite easily in order to operate with minimal interactions between groups. But thinking about going forward, we're scheduled to move into a new research building in a few years, and that really has me thinking about the new space that was designed there, because the same work that we're doing right now would not have been possible in the new in that new space. And so trying to kind of think, how would we deal with this if this happened 2 or 3 years from now in that new space, and how would we arrange things? We can't really change the architecture at this point for that new building, so we're going to have to be creative in other ways to think about how we would handle a similar situation. I think I can keep going on about other areas as well, but I think these are some of the important areas that kind of come to mind right now.
Thank you, Jeff. And how about you, Fred? Can you provide a key takeaway to share with our audience?
Yeah, I think I'm going to add on Jeff's comment. I think we've seen an amazing response from the scientific community. Openness, collaboration. I've seen private sector, academia, universities, colleges, anyone wanted to contribute in any manner they could, and that was awesome. That was an amazing time. I know it was a crisis, but at the same time, I think we saw what the community could do when we all put ourselves together and aim at the same goal. So that was, to me, really a great thing, and I hope that this spirit will stay a little longer. And I think we saw the potential of working together, and definitely for me, it's one key positive effect. Again, to Jeff's point, team is really important. We always say as a company that our team is the best asset that we have, and definitely in this type of crisis, we saw what our team could do, and it was tremendous. We were highly motivated. Everyone understood what was the goal. So it was an amazing time for our team to just put the pieces together, interact with collaborators and partners, and to find a solution. So there's a lot of positive things coming out of this crisis. There's another good side for me from this crisis, and that's very self-centered in some way. I do not have to explain what an antibody is. So that's a great thing for me. I used to spend at least a couple of minutes explaining the immune system and antibodies and what we had to do. But to be less personal and to a larger extent, I think the life science sector has been shown into another light with the public. And we saw scientists rise to the occasion and be really leaders in our societies in every country. And I think that's great news. That's great for us. But definitely we have to be now very cautious, very prudent. We've seen what accelerated research can do, the good side, but also some of the bad side, publishing very early on, taking every single study as facts. It has some advantages, but we need to make sure the quality of the research is to the standards that we usually have to go forward. But definitely, it's been an interesting time. Hopefully, we'll come wiser and more together than we were before. But we still have a lot of work to do.
Thank you, Fred, and also thank you, Patricia and Jeff, all of you, for this informative discussion. We will now start the live Q&A portion of the webinar. And just a reminder to our audience members, if you have any questions you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. So our first question coming in is for you, Patricia. For sorting of nuclei for COVID-19 study, should the processed sample be treated as BSL-1 or BSL-2?
Well, thanks for the question. Nuclei are typically pretty straightforward, and on an average day, we treat them as BSL-1. Now, for samples coming through that are COVID positive infected, you might want to take some extra precautions and sort them as BSL-2. Just out of abundance of caution, but it's more important of how you're dissociating the samples, because to isolate the nuclei, you often have to use ultracentrifugation, which can be very hazardous if you're using, higher biosafety level samples. And so wherever you're getting the samples from, you should use the appropriate biosafety level lab to isolate the nuclei. And then once you have the isolated nuclei, I think for our purposes, it would be fine to sort those nuclei in a biosafety level 2 lab. So a sorter that's inside of a biosafety cabinet. So yeah.
Thank you, Patricia. And this next question is for you, Fred. Are there any advantages or disadvantages, possibly, in screening COVID-19 antibodies using phase display versus using B cells?
That's actually a very good question. And the main answer is that both approaches have benefits and shortcomings. We preferred the B-cell screening approach for COVID-19, not because the phage display, we weren't confident in getting good antibodies from the phage display, but in terms of timing, the B-cell screening was a little shorter. We were saving maybe 1 or 2 weeks. So that's not a lot, but in this crisis, 2 weeks is a lot of time. But if I answer more generally, we typically, we sometimes do both for a single program. So definitely there's advantages, and good antibodies usually show up in both approaches. But let's say we are doing a program to find antagonist antibodies against a GPCR in oncology, would probably prefer a B-cell screening approach with a functionality assay. Whereas we have done very difficult programs where we know the immune response is very slight. There's not a lot of good antibodies. And one example that I have in mind is we did an RNA antibody, so basically an antibody that could recognize sequence specifically a small RNA. And you can expect from that example that there's not a lot of immune response against nucleic acids. So in that case, we use phage display because we're looking, we needed to enrich and to find these rare binders. And that's where the phage display platform is so powerful. So basically, in both cases, we would probably have found very good antibodies using either phage display or B-cell screening, but we took B-cell screening because we're saving 2 weeks.
Thank you, Fred, and I want to thank our audience for these great questions coming in. We have time for one more, and we'll turn to you, Jeff. Based on your experience so far, Can you comment on what type of flow cytometry assays are useful to monitor the efficacy of an antiviral therapy against COVID-19?
Yeah, this is a great question. So I think right now there is no great biomarker that would define a optimal therapy, whether that's a vaccine or a drug that would stop the virus. And so in light of that, what I'm seeing is people sort of going down two roads. One is looking at antigen-specific T-cell responses using cytokine flow cytometry to look at CD4 and CD8 T-cell responses to the peptides from SARS-CoV-2 and asking how many cytokines those antigen-specific T-cells produce, how broad is that response, meaning how many different peptides are being recognized. And then I think on the other side is the immunophenotyping, and this is looking at activation markers, the frequency of different immune cell subsets that are present in patients who respond better to the vaccines or produce a broader or larger number of antigen-specific T cells, or who suppress the virus more quickly on an antiviral therapy, and looking at those markers of immune activation, looking at the maturity of the T cells and the B cells, and probably we need to spend some time looking at the myeloid compartment and the NK cells too to understand what their role is in fighting this infection. But all of this really is aimed at high parameter flow cytometry. So whether that's doing something on a CyTOF or traditional flow cytometry with fluorescent markers, pretty much everybody seems to be going the route of high parameter. So we're in the process of making a 26 color, or actually pretty far along now in making a 26 color cytokine flow cytometry panel to look at antigen-specific responses, and then we have a 35 color panel that we're using to look at maturation and activation of the various different immune cell subsets, so we can look at T cells, B cells, NK cells, and myeloids in a single panel. And a lot of this is really aimed at screening patients and then understanding their disease trajectory or their vaccine response after 1 week, 1 month, 3 months, figure out how long these responses remain and to what magnitude they reach.
Thank you, Jeff. And I want to thank each of you again, Dr. Fred Laduke, Dr. Jeff Milish, Patricia Rogers, for your participation today and for your contributions to the scientific community. And I also want to thank Labroots and our sponsor, Sony Biotechnology, for underwriting today's educational webcast. Before we go, I also want to thank the audience for joining us today and for their interesting questions we did not have time for today, and those submitted during the on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. And this webcast can be viewed on demand. Labroots will alert you via email when it is available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, be safe. Bye-bye.